Stem cells in liver. So what are stem cells? Stem cells are the cells that proliferate and differentiate to form mature cells, and they do so under certain pathophysiological conditions. The stem cells are of three types. One is actual stem cells. It is based on regenerative capacity and the lineages. Actual stem cells are the true stem cells. And another is potential stem cells. These cells also have a good regenerative capacity. And the last one, that is committed stem cells. Committed stem cells are those cells who are committed for particular cell lineage, like stem cells in the liver. Now coming to stem cells in liver, they are of three types. One is hepatocytes itself. Second is stem cells in the canal of Hering. And the third one, stem cells from the bone marrow. Now coming to hepatocytes as stem cells in the liver, they are normally quiescent. That means they are normally dormant. They become activated under certain pathophysiological conditions, like after hepatectomy, and their differentiation option is only hepatocytic. That means they form only hepatocytes. Now coming to stem cells in canal of Hering. These cells are oval cells. They are oval cells. They are called bipotential precursors. They have a capability to differentiate into hepatocytes and bile duct cells. The ductular reaction we see on biopsy is because of these oval cells. And the markers for these oval cells are AFP, cytokeratin 19 and GGT. Now coming to the stem cells from bone marrow. It is actually the hematopoiesis in diseased states in adults, and they have a high self-regenerative capacity. And they differentiate into endogenous stem cells and liver parenchymal cells. Now coming to another topic, ITO cells. So what are ITO cells? We have number of names for ITO cells. One is fat storing cells or vitamin A storing cells or stellate cells. They constitute 5 to 8% of total liver cells, and they are present within the space of tissue, the space that is present between hepatocytes and endothelial cells. And what is the importance of ITO cells? They are the major site of vitamin A storage, and they produce extracellular matrix, which is important for scaffolding and cellular extracellular matrix interaction. They are also important for hepatic sinusoidal microenvironment, which is mediated through autocrines, paracrines, chemoattractants. In diseased states, these ito cells are activated and they transform into myofibroblasts. And these myofibroblasts are flat and elongated. Now coming to the morphological changes that are induced upon activation of ito cells. First is increased extracellular matrix production, and second is obliteration of space of DC. When there is increased extracellular matrix production, it leads to obliteration of space of DC, which leads to the defenestration of endothelial cells, and the process is called capillarization. As a result, there is a decreased transfer of solutes, and which results in hepatocyte dysfunction. Now coming to the organization of liver parenchyma. The major portion in the liver parenchyma is hepatocytes, and the arrangement is plate-like or cord-like, 1 to 2 cell thick. It is separated by sinusoids from portal tract to central vein. Now coming to the basic arrangements in liver. We have hepatic lobule, which is the main structural unit of liver. We have portal lobule, which is actually a model of bile acid secretion. We have hepatic acinus, and we have hepatic cords and sinusoids and hepatocytes. This is actually a diagram of liver architecture. In this you can see hexagonal cells with the central vein and portal triads. And you can also see a separate diagram of hepatic acinus. And in the acinus you can see 3 zones: periportal zone, and the periportal zone is the zone around portal tract. This is also called zone 1. In this zone you have numerous large mitochondria. That's why in this zone you can find there is increased oxidative activity. That's why the processes like glucogenesis, beta oxidation of fatty acids, amino acid catabolism, and ureagenesis and cholesterol synthesis, these processes occur in zone 1. Bile acid secretion also occurs in zone 1, that is the periportal zone. Zone 2 is intermediate zone. It constitutes features that has resemblance with both zone 1 and zone 2. Now coming to zone 3. Zone 3 has more endoplasmic reticular activity and microsomal activity. The processes like glycolysis, lipogenesis, and drug metabolism, that is drug detoxification,